Okay, so I'll go ahead and get started. Hi, my name is Hancock. I'm a PhD candidate in Texas A&M University, and I'm really excited to be here at Evolution. This is my first time, and I'm really excited to share with you some of the results that we have on a recent biogeographic study that we've done. If I'm bouncing around behind the podium, I'm sorry. I normally like to pace, but there's not a lot of room for it, so I'm just going to bounce around back here. So this project is focusing on the biogeography of sand-burrowing amphipods in the Gulf of Mexico. And these are particularly Haustoriidae amphipods. Now, when I say that to most people, it just makes their eyes glaze over. So a Haustoriidae amphipod is a type of brooding crustacean. When you see them, you would think that they kind of look like shrimp. And so what they do is, instead of just blasting their gametes into the water column, like most shrimp do, they actually brood their offspring. And so they keep them, the mothers will keep them on themselves, and these particular amphipods actually burrow into the sand. So they don't enter the water column, they're entirely blind, and so we think that they make ideal models for studying biogeographic phenomena. Now, for those of you that may not be too familiar with the biogeography of the Gulf of Mexico, there's a really interesting pattern of divergence in the Gulf. It's this east-west divergence pattern in sister taxa. Despite the fact that it's a contiguous ocean basin, right? You would think that these motile marine organisms would be able to traverse it quite easily, but yet we don't see that. We see lots of divergence in sister taxa. Now, there are many different hypotheses for explaining this, but the ones that I want to show you today are the two particular ones that we tested here. And so the first one is the runoff due to the Laurentide ice sheet. So this is a very shallow divergence. This one was postulated by Portnoy and Gold in 2012 to describe the divergence of lane snappers in the Gulf of Mexico. And so basically what you can think of this as is cold water came down the Mississippi River, and these organisms just physiologically couldn't handle it, and so they became divided on opposite sides. If this is the case, you would expect a pretty shallow divergence, right? Whereas alternatively, what we propose here is that you could also test with a bright model organism a much deeper divergence in the Gulf of Mexico. And so one of the events that was occurring during the middle Miocene is lots of sedimentation was pumping down along the Louisiana coastline due to the confluence of these three major river axes. So this is the Red River, the Mississippi River, and the Tennessee River. And if you are a coastal organism that relies on sandy substrate, then all of that silt and mud would be a barrier to your dispersal ability. So what we want to do is compare and contrast these two possible outcomes using these Haustoriidae amphipods. Now, we're looking at two distinct genera, so both of them occur in the Gulf of Mexico. One is Haustorius, and the second one is Lepidactylus triarticulatus. Now, what I want to show you is what the ranges of these two look like. So this is Haustorius. Now, on your left side is Haustorius galvezi. This one occurs across the western Gulf, and then on the right is Haustorius janii that occurs in the eastern Gulf. And this one kind of follows that expected pattern. You have the sister taxa divided on either side of the Mississippi River. Now, importantly, this is based on morphology. None of these organisms had any sequence data prior to this study. Alternatively, if you look at the distribution of Lepidactylus, it ranges across the entire Gulf, across the entire northern Gulf. It passes over the Mississippi, it crosses Mobile Bay. It seems to not have that same kind of distribution pattern. It's just one species across the whole Gulf. And so this leads to, and I should note that they have effectively the same lifestyles, right? They both live in the sand, they're both in the same family, and they're pretty difficult even to just tell apart. So this leads to a couple of questions. So what are the phylogenetic relationships of these amphipods in the Gulf of Mexico? Which of the biogeographic hypotheses that I presented before best fits the divergence times that we see or that we estimate? And do the two genera actually show similar biogeographic patterns or different biogeographic patterns, potentially indicating differences in life history or some other unknown variable that we don't know? Okay, so these are our sample sites. There's lots of little dots and boxes, but if you look around from 1 to 7 on the left, that's Haustorius galvezi. That's your western species. And then from 12, 14, 15, and 16 in green over here on the right, that's Haustorius janii. And so we got kind of the whole distribution of both of those species on either side of the Mississippi. And then 8, 10, 11, and 13 is Lepidactylus triarticulatus. And so we sampled that one again across its entire range. And so the hopes was that we would be able to capture populations on both sides of the Mississippi to see where those divergences are and if they show any clustering on either side. Okay, so what did we do? So we Sanger sequenced 4 genes, 2 mitochondrial and 2 nuclear, the CO1 and 16S mitochondrial, 18S and 28S is the nuclear. I'm just going to present a subset of the methods for time today, but we did TCS haplotype networks and pop art, and then we did phylogenetic analyses, including each gene considered separately, and then all of the data concatenated together using both maximum likelihood in RAXML or Bayesian analysis in Mr. Bayes. And then we did a dated species tree using StarBeast, and this is with the CO1 calibrated to that 1.5 to 2.5% divergence per million years estimate that you often see used in the literature. The reason that we had to do a molecular clock instead of fossil dating is because amphipods don't fossilize. We have no fossil data for this particular group, so we had to rely on the clock. Okay, so this is the CO1 haplotype network made in pop art. What I want to draw your attention to first is the ones on your left. This particular group here, so again, this is the map of their distribution, 1 through 7, and what you can see immediately is there's a ton of haplotypes, and that each of these groups have lots of genetic diversity and a lot of haplotype diversity. Another thing that you notice that's pretty interesting is that they're clustering north and south, and so instead of just showing that single panmictic population that we expected based on morphology, there actually appears to be a northern and southern clade that share no haplotypes, and they are diverged by about 3% at CO1. Now compare that to the Eastern Gulf, where you have relatively limited number of haplotypes, much lower nucleotide diversity, and it appears to be one panmictic population across the entire eastern coastline. Okay, so here is the full concatenated tree of Mr. Bayes. I've labeled this down from where the samples were collected from, as well as whether the clades are from the Eastern Gulf or from the Western Gulfs. That's the main thing that we're interested in here. And so what I want to focus on first is the Haustoriidae group. That's the one on the top. And so I'm going to put the map up down here on the bottom. So the triangles represent the Haustoriidae galvazi from Texas, whereas the circles represent the clade from Mexico, and then the squares represent Haustoriidae janii from that the eastern side of the Gulf. And so again, what we see is that same pattern. And then what I want to point out that's pretty interesting here is that despite the huge range of Haustoriidae galvazi in Mexico, right, it ranges from South Padre Island all the way to Campeche, it is divided from the northern coast by only about 100 kilometers. Those two populations are effectively abutting each other, and yet they don't share any haplotypes. So something is going on there around that particular part of the Texas coastline that is keeping these populations apart. Now let's look at the Lepidactylus clade. So this is the other one that was ranging across the entire Gulf. One of the things you're going to see here is the huge amount of divergence between these different populations, just looking at that phylogeny. So the circles and the triangle is the Western Gulf, and then the diamond and the square is the Eastern Gulf. And so each one of those individual sample sites represented a unique genetic lineage. It was at least 3% diverged from its nearest neighbor. And so they are not exchanging genes at all, and have not been exchanging genes for a particularly long time. And what we see is that they are indeed divided on either side of the Gulf of Mexico. Now, one of the things that you're going to note is that though there are 4 sample sites, there are 5 clades. And this comes about because here at the Diamond, this is past Christian, Mississippi, there's actually 2 distinct clades that we sampled at exactly the same beach and the same shovelful of sand. And they look identical. I could not tell them apart, and we never knew which one we were going to get until we sequenced them to see which clade they were going to fall into. And that corresponds to one that's actually closely related to the Western Gulf, though very deeply so. And then the other one is over here, and it's closely related to the Eastern Gulf, where that square at Pensacola Bay. And so that was completely unexpected and a pretty interesting finding. Now for the dated species tree, what this represents is so the width of each one of the branches represents the inferred N of E for that particular branch. And then you see 3 different trees that are layered on top of each other. The blue one is the most common one, and then the green and red being the 2 less common. And what you see here is the biggest kind of incongruence back in the past, for those of you that may be interested in the taxonomy of this group, comes about because of the Atlantic species. It's having trouble figuring out where they're clustering with. So the ones from the Atlantic are Canadensis and Arenarius that I've got shown up here that are sitting in between the other two clades, as it's having difficulty figuring out where those Atlantic taxa are actually related to the Gulf. Whether they're monophyletic with Haustoriidae, whether they're related to Lepidactylus, or whether they're just an entire outgroup to this Gulf clade. So that was pretty interesting, but for the dated part, what we're interested in particularly are these nodes, these ones with the stars. And so what we're asking is which one of these biogeographic hypotheses it fits into best. And so right here, highlighted in tan, this is our older one, right? This is the 5 to 10 million years during the Miocene. You had lots of sedimentation, and indeed, the 95% HPD on every one of these nodes falls within this range. Alternatively, if we look at the Pleistocene one, it should have been much shallower, right? The divergences should have been much shallower. Though we see the same species distributions we would have expected, they are far too diverged from each other to have reflected such a young event. Okay, so as I just mentioned, both of the genera show this east-west pattern of divergence. As I just mentioned, the timing likely reflects some sort of Miocene sedimentation event. And then we also see widespread cryptic diversity in this group. So Haustoriidae amphipods are relatively understudied, and they only have a couple of species in the entire family, whereas amphipods are known to be incredibly diverse. Just not a lot of people are working in this particular family. And so what I hope this shows is that there's a lot of diversity out there that we're just not seeing in these groups, and that without going in and sequencing these individuals, we're not going to know just how much diversity is actually hidden there. And so probably many of these taxa need to be taxonomically redescribed or reevaluated. And then I put a little check by the Miocene because that seems to be the best explanation that we have. Okay, so with that, I'd like to thank all of the people that helped me out on this project, particularly Faith Harden and Jessica Light. These are my two co-authors on this. They put in a lot of work. Neither of them knew anything about amphipods coming into this, and they were really interested and willing to learn, and I appreciated that. My advisor, Mary Winston and Anya Schultz, that helped me get down to Mexico and sample, and then all of these other great people that helped out holding buckets and shoveling sand across the Gulf of Mexico. I could not have done it without them, as well as my funding agencies, Texas Eco Labs and Sea Grant, and my program provided a grant as well. So there is a lot of things that I didn't get to present today. If you're interested in seeing more of the results, particularly more of the PopGen stuff, please come by my poster. It's 1:32 on Monday, and I'd be happy to take any questions.
I mean, my first impression is I'm surprised that you were able to recover all of this structuring in the data when you consider. When I think of these beaches, I think of erosion mitigation and people trucking in sand. Did you account for that, or how could that be influencing what's going on?
So if that was the case, if that was what was dividing them, you would expect, like dividing populations, for example, then you would expect the divergence to be really, really shallow, right? Because this is something that's only been happening for 100 years or so, whereas these divergences are incredibly deep. Now, interestingly on the beaches that have been heavily raked or manicured, you don't find amphipods. They're not there. And so I have to go to state parks, I have to go to protected beaches that you don't find lots of tourists on. Sometimes you can find them, but it takes a long time and that they don't flourish in those beaches.
And maybe those areas haven't been mitigated as much, so you don't have as much sand mixing around?
Perhaps. Yes?
How did you account for the tendency for mitochondrial data to overestimate divergence time?
To overestimate divergence time. So the difficulty here is that that's largely the loci that had the most information, right? So what I did find is that both 18S and 28S showed the same clades, and they actually, when I did the star beast analysis based on just the CO1 alone, the CO1 alone definitely gave a much deeper time even than what these showed. But when I did the species tree approach and actually used the information from all the other loci, it dragged that time out quite a bit. And so I'm hoping that going that route at least attempted to account for some of that. But yeah, that's definitely a possibility that I wasn't able to cover all of it just because I have a few loci.
Yeah, even multiple mitochondria
Sure.
divergence.
Yeah.
So do you have any plans of including some of
Yeah, so we're actually in the process of preparing RADSeq libraries to redo this whole thing with genomic scale data, and then we'll see how the divergences look.